Sí,
Bueno,
sí.
pues hoy tenemos el gran placer de tener como invitada a la doctora Constantina de la Universidad de Rockefeller, donde es investigadora postdoctoral en el laboratorio del Dr. Jarvis. Quisiera decir que también que la invitación a Constantina fue por parte de una recomendación de un estudiante del posgrado. Entonces, si tienen recomendaciones para los próximos seminarios, adelante. La doctora Constantina usa genómica comparada para descifrar la historia evolutiva de familias de genes y su propósito es establecer una nomenclatura universal para todos los vertebrados, para los genes de todos los vertebrados. Es un miembro activo del Vertebrate Genomes Project, cuyo propósito es generar genomas de referencia casi sin error para los casi 70.000 especies de vertebrados, que es una meta muy ambiciosa. Para su doctorado, realizado en Barcelona, Duke y Rockefeller, se enfocó en el papel de la oxitocina de Rockefeller en China sobre la evolución del comportamiento social en humanos y la evolución de la comunicación vocal, utilizando a las aves canoras como modelos, y sobre la influencia de la domesticación sobre los genomas de las especies. Entonces, pues le damos las gracias a Constantina por este seminario y the floor is all yours.
Thank you. Thank you for having me, and many thanks to the students for inviting me and for picking me and for the invitation. It's really great to be virtually in Mexico. So today I'm going to present several lines of research that all have in their heart my biggest passion, which is vocal learning. Give me a second. Okay. So I think I've got this now. Good. So vocal learning, as I said, is my big passion and where everything started off from. And with this term, I mean the ability that we think many humans think we only have, and that we term it as speech or language, but actually there are many more species that have this ability to learn and imitate complex sounds, some of which you can see on my slide. For example, among mammals, it's not only humans, it's also elephants, pinnipeds like sea lions, or bats and dolphins. And from the songbirds we have, and from the avian lineages we have songbirds like the zebra finches that you can see here, or hummingbirds and parrots. So although these species are considered the ones that show this ability that I call here vocal learning in a fully fledged version, of course there are other species that also communicate through vocalizations that are really worth studying. So what really has attracted my attention from the beginning has been the fact that social learning actually happens in a social context. Vocal learning is something that, speech learning, as we call it in humans, is something that's happening necessarily with your parents or your caregivers. We cannot think of vocal learning as something happening outside of a deeply social context. And this is something that through studies we have come to understand that it's also happening in several other vocal learning species like the zebra finches that we see here. So although it seems something that's really essential for vocal learning to happen, this social reward or social motivation mechanism, actually there is no one that has studied really in detail whether indeed social reward affects and to what extent it affects vocal learning, or would vocal learning be possible without social reward, etc., etc. So I wanted to deep dive in this question and see where social reward and vocal learning touch. So the experiment that we set up in order to study this question goes a bit like this. So we used zebra finches as model animals to study this behavior, and they were baby zebra finches. They were infants, and the first thing that we did was that we wanted to train them to sing a song that we would use as a basis in order to afterwards test whether social reward can have an impact on it. And so the basic song that we had them learn in the first 65 days of their lives sounds like this. So you hear that it consists of 2 syllables that they are getting repeated several times, but it's a pretty simple song, someone could say. And in the first, from the day 30 to 40 in their lives, they were just sitting in their boxes with a model animal, which is a wooden animal that looks like a real animal. And at least from other studies, we know that they consider it as being a real animal. And from the loudspeakers, we have this song played several times throughout the day. When they reach the day 40 of their lives, we add these little buttons, these little keys that they can peck on in order for the sound to get played from the loudspeakers, because earlier experiments have shown that in this way they are more attentive to the sound produced by the loudspeakers. So this is how the experiment goes. As I showed, first passively and then actively, they're trained to learn this basic song, and then from day 60 is when the fun begins, because we change them every other day to these different boxes, the one being what we call a social reward context and the other being an isolation context. In the social reward context, we had the bird be with his model wooden father in the same box, and also with a female. And in zebra finches, it is important to say that the females are not singing, so they're not really intervening with what's happening, with the vocal learning that's going on. And we also added a mirror that in other experiments, it has been shown that it gives the impression that there are more birds around it. So in this controlled but still social rewarding context, we were playing a specific kind of song that I will play just afterwards. And then in the isolation context, we were playing a different kind of song. Actually, the difference was that in the first song that I played, in this basic song, we were sifting by 2 semitones, the second syllable of the song. Either we were sifting it up or down. So if the basic song sounded like this, we were then exposing them to either this song that was sifted up or to this song that was sifted down. And so every other day we were changing them. So let's say that in day 61, the bird was put in a social rewarding context, on day 62 in the isolation context, day 63 social reward context, and so on and so forth. And we were, of course, changing the song. So we were not always adding the same kind of pitch in the social reward context for all the birds, in order for us to make sure that the choice that the bird would make would be just because of the context, and not because just randomly they preferred singing the one song versus the other. So our results was that 5 out of 6 birds learned the pitch that they were listening to while they were in the social reward context, whether this was a sifted down pitch or a sifted up pitch. So this gives some first evidence that indeed social reward gates actually some specific aspect of vocal learning that we studied, which in this case is pitch learning. So since I have talked about how social reward interacts with vocal learning, then we wanted to delve into the mechanisms, the brain mechanisms that make this social reward pathway possible in vocal learners. And so what we hypothesized was that oxytocin could be playing a very important role in the social reward brain pathway, because we have put together in these papers several experiments that are showing, for example, that in the human laryngeal motor cortex, that is very important for the production of vocalizations and speech in humans, there is, for example, a specialized upregulation of the expression of the oxytocin receptor, or in similar respective regions in the songbird, and several other lines of evidence convinced us that actually oxytocin could be a very good candidate for the study of social reward in vocal learners. But then a problem came up. The problem that came up was that several colleagues of ours, or even reviewers of these first papers that I showed, they were telling us, well, you cannot be saying that you're studying oxytocin in birds, because birds do not have oxytocin. They have something similar, but it's not oxytocin. It's called mesotocin. So you cannot be saying that you're studying oxytocin in songbirds. And we said, wait a second. What is going on here? Why is it that birds do not have oxytocin? So they don't have the gene, or they have the actual gene but with a different function? So this is a bit confusing. And it got even more confusing when we were trying to find the sequence for the oxytocin receptor in birds, and we were not even able to find something that was oxytocin-like, like an oxytocin-like receptor. We were finding receptors named vasotocin receptor 3, 4, 2, 1, but no oxytocin receptor. And then we decided that we really need to resolve this question in a firm manner, and in a manner that would really help the researchers in the future not to have similar questions and similar confusions. Because as we realized, this is not a problem that's going on only when someone wants to study birds versus some other mammalian species, but also in all these other species, someone can find a similarly confusing nomenclature. So you can see here, for example, how many names we have for what we call in mammals oxytocin: valitocin or aspartocin in sharks, isotocin in fish, and a similar chaos is going on for the several different receptors. So with the analysis that I'm going to show to you, we try to propose a nomenclature that would be universal for all vertebrates for these genes. But as I said, according to some of our colleagues, gene nomenclature should be based on function. So since there is a gene, they're saying that it has some sequence similarity with oxytocin with the mammalian oxytocin in birds, but since it has a different function, we should call it differently. And so our reply to this is that if you only take some examples of the functions of oxytocin in several vertebrate lineages, you will see that actually there's so many different functions that it has within a lineage and across different lineages, that if we were coming up with a different name for different functions in different species, this would become a really chaotic situation where we would have to have really too many names, and the communication would be really, really difficult. And you can see, of course, that there are several similar functions across different lineages, but also some functions that are really lineage or even species specific. According to other scientists, gene nomenclature should be based on the sequence identity between paralogs, which is something that was happening a lot in the pre-genomic era. Namely, someone was finding a gene, and some other scientists had found some genes, let's say in the mouse, and they were comparing these genes for sequence identity, and they were finding that, okay, it looks more identical to that gene in mice, so okay, we will call it as it being this gene. And so what we realized was that actually, although this would be the expected outcome, namely that one receptor from each species would look more like the respective orthologous receptor to another species, we found out through long Excel tables that we run that actually this is not the case. Because, for example, you can see here the C. lamprey oxytocin receptor, that it doesn't match in terms of identity best to the frog oxytocin receptor. It matches best to the frog vasotocin receptor 2C. So again, sequence identity is not a measurement that is good enough, that is solid enough, that is helpful enough for us to define a gene nomenclature. And according to us, gene nomenclature should be based on what we call synteny. So what is synteny? So when we have a favorite gene and we want to study its function in several different species or in several brain regions, for example, what we usually do is that we go to the genome of to the genome of the species that we study. For example, here we have the human genome. We search for the oxytocin receptor, and what we're really interested into is the sequence of the oxytocin receptor, and we don't look anywhere around this gene. But actually, synteny is this territory, all the surrounding genes that kind of embrace this gene of interest, which is the oxytocin receptor. Namely, what makes a gene what it is not only what is inside the gene, but also what it is outside of the gene, namely the genes that surround this gene of interest. And here you can see, for example, a 10-gene window synteny for the oxytocin receptor in several species that represent the main vertebrate lineages. But we actually did all of these analyses in 35 vertebrate species and 4 invertebrate outgroups. And here you can see, for example, that the oxytocin receptor, the human oxytocin receptor, has a specific kind of synteny, has a specific kind of territory, of gene territory, that is just unequivocally similar to the oxytocin receptor territory of chicken. And so easily, even from a 10-gene window synteny, we can clearly find which gene is orthologous, truly orthologous to which. And of course, as I told you before, this gene in the current genome annotations is written as mesotocin receptor or vasotocin receptor 3. Based on this synteny analysis, we can understand that it is actually the orthologous oxytocin receptor, and there is no reason why we should call it with a different name. But of course, a 10-gene window synteny sometimes is not large enough to show the true orthologous relationships between, for example, some teleost species that have several rearrangements and translocations. So we really need larger synthetic windows in order to understand these true orthologous and paralogous relationships. For this reason, the high quality genomes that the Vertebrate Genomes Project has been proposing and actually making itself have been very, very important. So with this new combination of technologies that we wrote about here recently, we are putting forward a technology where we would have long reads as the building blocks of the sequencing, and then through other technologies through other scaffolding technologies and different annotation annotation runs, we would have a really high quality assembly. And why is a high quality assembly so important for this kind of analysis that I presented here? So first of all, both this study for the Vertebrate Genomes Project and the study of the oxytocin receptor, they were both featured in a special issue for the Vertebrate Genomes Project in the end of April in Nature, and we have this nice Anna's hummingbird in the cover that we sequenced in high quality. And so, but a specific example, why is high quality really important? So for this synteny analysis, we can see here that in this in the genome of the platypus, which is this beautiful mammalian species, the vasotocin receptor 1A, which is what was previously called in mammals vasopressin receptor 1A, we can see that if you actually wanted to search for this gene, you would find it as an only gene on a scaffold. Namely, you wouldn't find any genes before or after this gene. But in the new assembly, actually all the genes around this gene of interest are found on its chromosome that is fully assembled and annotated, something that can let us do well this synteny analysis, because with only one gene, you cannot run any synteny analysis. And so we did this more macro synteny, this further larger synteny analysis using not 10 genes, but up to 100 genes in order to see, for example, is this gene in humans mostly synthetic to which gene in other species like chicken? And here you can see that this gene, which we end up understanding that it is the human vasotocin receptor 1A, is mostly orthologous with the expected vasotocin receptor 1A in blue in chicken. Although, of course, it has some genes in common in its synteny with the other paralogous genes of the same family. We also wanted to do this in a larger chromosomal scale in order to find whether this is only a synteny that stands in a 10 gene window, in a 100 gene window, or in a larger window synteny. So, and we've really found that these scaffolds, for example, in the sea lamprey, where we can find the oxytocin receptor and the vasotocin receptor 2B, that they have significantly more synteny with these chromosomes in the medaka genome, in the chicken genome, in the frog, in the human genomes, where these same genes, the oxytocin receptor, for example, here, and the vasotocin receptor 2B are located. Something that shows to us that actually the orthology is not a short scale orthology, it's an orthology even at the chromosomal level, something that supports a deep ancestral paralogy between all these chromosomes, suggesting that they all happen through some duplication phenomena. And this is something that we also wanted to study within a species. Namely, is it that we are finding these synthetic territories, synthetic chromosomes only across species, or we would find them also within species? And we found, studying the human genome, that of course there are several other paralogous gene families around these receptors of interest. Like here you can see, one gene family with 3 or 4 genes being around these genes of interest. And in this way, through all these micro and macro synteny analysis, we were able to finally understand which are the genes that exist in which species and which lineages, which are the genes that have been lost, etc., etc. And in this way, of course, a suggestion, a proposal for a universal vertebrate revision was made possible. And so we really propose that we scientists can speak the same language by using the same name, at least across vertebrates. And if you can see here, when we took the names that we understood through synteny and we mapped them on top of the 3 leaves of the phylogeny, we can see that actually it maps with an almost one-to-one consistency, because you can see here, that all the vasotocin receptor 1A is mapped together and they branch together, and all the genes work out actually pretty well in the phylogeny compared to the old phylogeny. And here, not all the trees are expanded, which was pretty confusing, because, for example, here you can see that the same gene name was being used for what was different genes in the same species or in different species, or for what is the same gene, you can see different names being used. So at least with our new nomenclature, all these confusions and inconsistencies could be resolved, which is, I think, a good progress for science communication, at least. So what happened with oxytocin in terms of evolution? So in the jawless vertebrates like the sea lampreys and the hagfises, we found only a gene that we infer was this vasotocin ancestral gene. And in the jawed vertebrates, starting with the genomes of the elephant shark, the coelacanth, and the others, we found next to it the gene that we inferred was oxytocin. There were other people before us who had hypothesized that actually oxytocin is the newer gene, the one that got duplicated. But it was us who first showed evidence of why this is the case. Namely, why isn't it the mother gene oxytocin and this duplicated vasotocin? Why do we say that this was the first gene? And so we found around this oxytocin gene DNA transposable elements that according to other studies, they have been found to drive local duplications. So they start copying and pasting whatever they want to just translocate and copy and paste with these transposable elements starting and finishing the duplication. And we found also some other features, like, for example, a higher GC content in the intron of the oxytocin compared to vasotocin, that is also suggestive that this gene is the newer one. And so we propose that we call these genes oxytocin and vasotocin, because through this ending tocin, we can show that they are paralogous genes, and that if they were named today, probably they would be called something like vasotocin 1, vasotocin 2, or oxytocin 1, oxytocin 2. But we cannot change the nomenclature probably so dramatically. So we're proposing this change as a less dramatic one, but still that it is more helpful and it portrays the evolutionary history. And for the receptors, what we suggest is that they probably came about through one round or two rounds of whole genome duplication. The one round is explained as follows. We found in the vertebrates one vasotocin receptor, and then we found in the hagfish two vasotocin receptors. So we hypothesize that probably they got segmentally duplicated in a way that reminds us of this duplication that I just explained for the oxytocin and vasotocin ligands, and that then these two receptors got duplicated through one round of whole genome duplication, and that they gave rise to 4 receptors in the lamprey ancestor, and that then through some specific segmental duplications, these other 2 genes came about. The story that the traditional scenario was that there were actually 2 rounds of whole genome duplication that led to the genomes, the vertebrate genomes that we see today when we sequence some vertebrate genomes. But actually, what we put forward is that with 2 rounds of whole genome duplication, we would have to suppose that of the 8 genes, 2 genes got fully deleted at some point in the jod vertebrate ancestor. And we didn't really, of course, find any sign of this gene in any vertebrate genome of the genomes that we tested. And more importantly, we did not find any sign of synteny around these genes of interest that would show that probably at some point this gene existed, but its synteny has been preserved. So that's why, although of course we're not excluding this scenario, we say that this scenario is probably more likely. So after we've clarified the orthologous relationships between the oxytocin, vasotocin ligands and receptors across species, and we can happily say, yes, we're working on the oxytocin system in the songbird, I'm able now to show to you what we found that the oxytocin is doing in the songbird brain in adult zebra finches. So what we did is that we intranasally administered an oxytocin antagonist in the zebra finches, in the male zebra finches, who are the ones who sing. And we wanted to study the changes of the song that they sing to their females in order to attract them. So something that has been found in other studies that is really particular of this sexy song of the zebra finches to their females is that they are adding several introductory notes in order to attract their attention before they sing the main song. But what we found when we treated them with an oxytocin antagonist is that these introductory notes really got reduced in a way that was significant, something that suggests that at least in adult birdsong, oxytocin is playing a role in modulating specific aspects like the introductory notes of their singing to their females. So I've spoken so far about how social reward and how social learning in general interacts with this vocal learning module, as if it were, although I don't really like this word. Now I really want to take this thought in an evolutionary scale and see whether social evolution can be shown to interact somehow with vocal learning evolution. And in order to do that, I will try to shed light to this connection through studying a specific hypothesis of social evolution in humans and, of course, other species that is called the domestication process. And in humans in particular, it has been called self-domestication. So probably it sounds pretty weird, but what does this mean? I'm sure that we all know and we all understand that when we talk about domesticated species, we mean that we speak for species that are tame, for species that probably have been selected for in some way for their behavior. But actually what has been found and has been observed not only by Darwin from a very beginning, but now from several other scientists, is that this selection for a more tame behavior actually comes together with several other characteristics that could be thought of as byproducts, which could be curlier tails, shortened snouts, a different brain size, reduced teeth size, pigmentation changes, and floppy ears in the domesticated counterpart compared to the domesticated species. And why do I say that we could shed light to human sociality through something that we call self-domestication? Because actually, if you compare the skulls of the Neanderthals that have been found, that can be thought of as being a good proxy for our ancestors. If we compare them with the modern human skulls, we can find that first of all, the skull shape and the size is different. The modern human one is smaller, something that we also find in the domesticated species. Same holds for the brow ridges. The brow ridges are really more protruded, expressed in the Neanderthals compared to modern humans. Same holds for the nasal bones, for the teeth, and for the jaw. This is a really more protruded jaw compared to the human one. So although this is a hypothesis that has been put forward in the past, we really wanted to see whether in the genome we could indeed find something that could support this hypothesis. So what we did was that we wanted to find whether regions under positive selection in the human genome would overlap with regions under positive selection in the genome of other domesticated species. And what we did was that we compared these genes that were under positive selection, and we wanted to find whether this overlap of genes under positive selection in humans and other domesticated species was statistically significant. And we found that with the overall set of all the genes under positive selection in all these species, domesticated species that we studied, like cattle, cat, horse, and dog, was significant. And also if you compared the genes under positive selection only in humans versus cattle, there was a statistically significant overlap, as well as if you did the same comparison with modern humans and dogs. And so this is the first genomic evidence that shows that, well, probably this self-domestication hypothesis for human evolution is not something that only can be supported from these similarities in the face, but also through some genomic data. But of course, this wouldn't be enough if we didn't show a more controlled test, which would be, right, if humans show some statistically significant overlap with these domesticated species, what about other non-human primates? Do chimpanzees, orangutans, or gorillas show any significant overlap in their genes under positive selection with domesticated species? And we found that none of them does, which really suggests that it is only humans out of these species of these great apes that we tested that shows something similar in their genomes with these domesticated species. And although I don't really like genontology analysis, we found that this genes that overlap in humans and domesticated species, that they're involved in neurocrest formation that has been repeatedly mentioned in studies about domestication, in neurogenesis, neuronal development, and synaptic plasticity, and also mutations in these genes give rise in humans to autism, schizophrenia, and Down syndrome. And so, since I'm trying to speak about social evolution, and in particular about the self-domestication hypothesis, I also want to show and explain why oxytocin is a good candidate, not only for the social reward of vocal learning, as I said before, but also for our social evolution in general and for self-domestication in particular. Well, for our social evolution, I think there is no wonder, because there are so many studies that have shown that oxytocin is involved in labor, in breastfeeding, in social bonding, and so on and so forth. And but in terms of self-domestication, it has been shown that actually there are changes in the oxytocin expression in the brains of domesticated versus wild rats and mice. Same holds with specific oxytocin receptor differences in the sequence and methylation patterns in dogs versus wolves. And even we know of so many studies that show that there are differences in the oxytocin levels when we spend time with our pets. So what I really want to explore here is whether oxytocin can shed light to social evolution and probably even self-domestication. So jumping off again from this comparison, we really wanted to compare the sequences of the oxytocin receptor, the vasotocin receptor 1A and 1B, between modern humans and Neanderthals, as well as Denisovans, in order to see whether they can give us any hint of what was happening in the oxytocin and vasotocin system, and whether these can underlie their differences. So we compare the full genomic sequence of these genes, and we also compared these gene sequences with the same sequences of 12 non-human primate species. And when it comes to chimpanzee, bonobo, and macaque, we had actually 3 to 1,000 individuals per species. So we could account for variation also in these species. And we used only those sites in the modern human genome that were already heterozygous, because it's mostly for these sites that we have information for whether this or the other site, or this, or this site as a whole, has any impact to social behaviors. So we found several spots in the genome that have either a modern human unique variation, or where this site is variant also in other species, but the specific variation is modern human specific, or other variation patterns that we share with archaic humans, etc., etc. And what we did also was that out of these sites that we found, we wanted to see whether they are functional in terms of open chromatin data, or even in terms of whether they are found in sites that are transcription factor binding sites, etc., etc., or whether they're active in the brain in specific brain regions. So based on this, what did we find? So we found five sites with modern human specific variation, where the modern human allele is the major allele in the global population. We found 2 sites on the oxytocin receptor that are present only in modern humans and archaic humans, and they're under positive selection in modern humans, and one of them is very highly functional. And we found 3 sites also, interestingly, of convergent evolution between modern humans and bonobos, because as some of you might know, bonobos have also been found or hypothesized to be a self-domesticated species. And out of these sites, one of those that are on the oxytocin receptor are highly functional in terms of them studies that I mentioned before, like whether they are active in the brain and whether they are found in open chromatin sites, and they're under balancing selection in modern humans. So now that I tried to create a link between social evolution and probably self-domestication and oxytocin through this study, I also want to bring in, to make a full circle, vocal learning evolution. And there is a specific pair of species that makes this very easy for me, because these species, so this is this Bengali species that is the domesticated counterpart of the white-rumped munia. And they were imported about 250 years ago from China to Japan, where these wild birds, they got selected for, you know, the ones that were the least aggressive. And then we have, as if it were suddenly, but it's not how it happens, we ended up today having this population of the Bengalese finches. So they show several of the characteristics that we find in mammalian domesticated species, like feather color changes, or of course similarities in behavior, because these species, the wild type, is more aggressive, it bites more, it has a higher stress reaction. But the interesting thing is that they have a big change in how they sing. So these species, the wild species, really has a simpler stereotyped song. But when you select, and you select only the least aggressive birds of this species, what you get in tandem is also a more complex song that has larger note-to-note transitions, and also higher pitched notes, etc., etc. So this gives me the great opportunity to be able to see how social evolution, and particularly domestication, interplays with vocal learning, and how oxytocin could subserve all this game here. And so what we did in collaboration with Yasuko Tobari and other members of the Kazuo Kanoya lab is that we studied whether we would find any differences in the oxytocin gene of these two species or populations, and we found a decreased synthesis of oxytocin in the domesticated counterpart. So actually, first of all, we did an oxytocin gene sequence comparison, where we found several places, locations in the genome of these species where they both differ. And we found that, and we also compared these loci to see how they look like in other avian species and also in other vertebrate species. And although, of course, we cannot say something like, oh, we found something that only the Bengalese finches have and no other species in the animal kingdom, at least we were able to narrow down to some hotspots where probably oxytocin is working in a different way in these two different species, probably to underlie some of the changes in their behavior. What we found with real-time quantitative PCR is a decreased synthesis of oxytocin in the Bengalese finches, namely in the domesticated bird. And we didn't find in the diencephalon, namely in the hypothalamus, thalamus, epithalamus, etc. But we didn't find it in the rest of the cerebrum. And this really surprised us, because, as I said before, in mammals, we actually find an increased oxytocin expression in domesticated rats and mice. And we found decreased oxytocin expression in the domesticated Bengalese finch. And so this could be, of course, due to there were several different mechanisms through which oxytocin might be working on, and it doesn't work by itself. So it works in tandem with so many different systems. So of course, finding a decreased synthesis in the one versus an increased in the other probably doesn't say much. But one way we could interpret this finding is that these two species went through a different pathway to reach their domestication. So what we know about mice and rats is that they were acclimated to humans. They knew what humans were, and they were acclimated to their presence in the 17th and 18th century. And then this really lab domestication, which is more intensive, took place. Whereas in the case of the white rump munia, they didn't have this kind of acclimation. And as I said, they got imported to Japan about 250 years ago, and there was a specific selection for the least aggressive individuals that gave rise to this domesticated population. So of course, it could be because we're speaking about different lineages. It could be because we're speaking about different pathways that led to domestication or other mechanisms. So what I tried to do today here is to show how in development, social learning or social motivation, social reward actually can have an important role and can help us understand vocal learning and what's going on while we learn how to speak, or for other species, how they learn how to sing. And what's the role of oxytocin in this interaction, both, as I said, in a developmental window, but also in an evolutionary window, and taking as a specific hypothesis of social evolution, the domestication and self-domestication in particular for humans. And so the conclusions for the several of the studies that I have presented are that social reward gates vocal or at least pitch learning in zebra finches, that we have some preliminary results that there is a role that oxytocin is playing in underlying birdsong features like the introductory nodes in zebra finches. We are very happy to have put forward and have found a synteny-based nomenclature for oxytocin and vasotocin ligands and receptors across vertebrates, something that has made all this research possible. Although it seems really like that we changed gears in the on the way, it was really important for us and probably for the future generations to be able to study the oxytocin system well. And we found, in terms of the self-domestication hypothesis in humans, a significant overlap in the science of positive selection between humans and other domesticated species like the dog and cattle. And in particular, in this link between oxytocin and social evolution, we found that there are modern human-specific or modern and archaic human-specific variants, or even variants that are convergent and exist only in modern humans and bonobos in the oxytocin receptor, vasotocin receptor 1b genes that can be used as a window for further studies to study how these specific loci might be having a role in social behavior. And we found also a decreased oxytocin synthesis in the domesticated Bengalese finches that can have, as I said, many interpretations. So I think this is it, and thank you very much for having me. I really want to thank my mentor, Eric Jarvis, for all his support throughout these years, and all my collaborators, and without them, of course, it wouldn't have been possible. Thank you very much, and I'm open to any questions you might have.